And I don't know, this is just my feeling, you know. It seemed like we were being offered two alternatives. Use methyl iodide and jeopardize the health of workers and their families. Use methyl bromide and deplete the ozone. The third possibility is you don't use methyl iodide or methyl bromide and you make strawberries a little more expensive. And I don't know, but when you're left with these kinds of serious issues, depleting the ozone, hurting workers and people who live in the fields nearby by adopting a standard that makes no sense whatsoever, that third option begins to sound like not unreasonable. Mm-hmm. Let's just charge a little bit more for strawberries. Mm-hmm. If people understood, I think they might consider that. Well, I do think I've gone on too long, so let me
well, thank you. Thank you for your comments, and thank you for traveling here. Okay.
Am I on next?
Ted?
I want to thank Senator Flores and the other members of the committee for enabling me to fly cross country and take the red eye back.
Thank you for coming.
My name is Theodore Slotkin. I have a Ph.D. degree in pharmacology and toxicology from the University of Rochester School of Medicine, and I've been on the faculty at Duke University Medical Center for over 40 years. I hold full professorships in the Department of Pharmacology and Cancer Biology, in the Department of Psychiatry and Behavioral Sciences, and in the Department of Neurobiology, which shares the blame. And I'm the Director of Graduate Studies for the Integrated Toxicology and Environmental Health Program. I sit on the editorial boards of 4 scholarly journals, including Environmental Health Perspectives, which is published by the National Institutes of Health and is the top-ranked environmental health journal in the world. I've authored over 500 peer-reviewed scientific articles. I'm among the top 5% of NIH grant recipients over the past 25 years. And I'm ranked as one of the top 1% of cited scientists in pharmacology and toxicology. On this review panel, I was asked to review the effects of methyl iodide as they relate to 3 specific areas in which I have expertise. And it's going to be easier because I don't have to give you numbers because I'm going to point out that the data are just lacking for these important areas. Neurotoxicity, which is toxicity directed towards the structure and function of the brain. Developmental toxicity, which means the enhanced sensitivity of the fetus and young child to toxic chemicals. And the combination of those two, developmental neurotoxicity, which is the special vulnerability of the developing brain to toxic insult, the sorts of things that lead ultimately to neurobehavioral deficits, attention deficit, autism spectrum disorders, lowered IQ, all of the things that I'm sure you're aware of. Because the details of my conclusions are in the report, I'm just going to focus on the essential meaning of those conclusions and skip the details. And I'll deal with them in sequence. First, there's absolutely no question that methyl iodide is neurotoxic. If you look at the material safety data sheets that are provided by companies that manufacture methyl iodide, or the HAZMAP listing provided by the National Library of Medicine, it says, and I quote, "Central nervous system is the target organ." Points out the emergence of, quote, "chronic neurologic symptoms that do not become manifest until days or weeks after exposure," end quote. Mm-hmm. The case studies of people after methyl iodide poisoning all indicate lasting neurological damage, including severe psychiatric symptoms and movement disorders that resemble Parkinson's disease. Laboratory studies similarly show clear-cut neurotoxicity, and they've identified the specific parts of the brain, types of cells that are targeted. And on top of that, methyl iodide concentrates in the brain. So if you do safety assessments that are based on blood levels or inhalation levels, the brain is actually experiencing a much higher exposure than you would expect from any of the standard kinds of measurements. So what I'd like to do is to address the issue of the fact that the neurotoxicity, quote, studies that were used by DPR and provided by Arysta were nothing of the sort. They were not neurotoxicity studies, and they were totally inadequate to provide a launching pad for even beginning to calculate safety factors. The exposure paradigm that was reported to DPR by Arysta and used in their assessment was limited to a single episode of exposure, and the behavioral tests were conducted during and immediately after exposure with a single assessment, very crude assessment of structural changes 2 weeks later. Let me give you an analogy. Suppose I wanted to study whether chronic alcoholism damages your brain and your behavior. And I think we all know that it does. And I'm going to do that by getting you drunk once, measuring how long it takes you to sleep it off, and asking you 2 weeks later if you feel okay. I think I would conclude that, hey, alcohol, no problem, right? That's the kind of study that this was. That is not a neurotoxicity study. Beyond that, this basic design defect, the techniques that they used came out of a paper that's 25 years out of date. All right? No one in developmental neurotoxicity or neurotoxicity would use these techniques. They're insensitive. They're inadequate. The company that they hired to do this provided background data of supposedly positive control studies that they did, and they used a vicious neurotoxin, trimethyltin. All right? Most of the animals, they couldn't detect anything. They had a higher rate of false negatives than ones where they could actually detect anything. This is to give you an idea of the inadequacy of the launching pad for even beginning to use a number and calculating a mitigation factor based on it. Based on these kinds of studies and this approach, you would also conclude that lead, mercury, organophosphate insecticides, and alcohol are not neurotoxic. In the absence of a chronic exposure model and a properly conducted neurotoxicity study, there is no way to establish a no adverse effect level for the neurotoxicity of methyl iodide, and it's certainly going to be much, much lower than that which was postulated from the study that was provided. Number 2, developmental toxicity. Well, here the most sensitive endpoint was fetal death. And, you know, it's already been pointed out, fetal death is the crudest endpoint you can imagine. So short of fetal death, there's going to be all kinds of other stuff happening. I'm not going to go into more detail as to what was wrong with the studies that were provided. They were based on the supposition that fetal death had to do with effects of methyl iodide on thyroid function. You have my written comments. That's just wrong. Arysta's own studies showed that that was the wrong assumption. I had to point that out to them during their hearings. They seemed confused by it. They made an excuse that methyl iodide was more thyroid toxic than sodium iodide, the supposed negative control. And I pulled the data out during the hearings and showed them that they were identical. They were supposed to get back to me about it and never did. All right, so the conclusion is it's fetotoxic, all right, at levels much lower than that are toxic to the adult. We don't know why. It's clear that using fetal death as an endpoint is ridiculously overestimating where the level should be, and we have no way of knowing where the level should be because we don't know the physiological changes that are contributing to the death and that are undoubtedly happening at much, much lower levels. So now I'm going to combine the two, neurotoxicity, developmental toxicity.